Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For the section to discuss NCRT Class 12, the chapter Biotechnology and its applications. Part 4, Molecular Diagnosis. Molecular Diagnosis. You know that for the effective treatment of disease, early diagnosis, that is understanding its pathology is very important using the conventional methods of diagnosis like as in serum, urine analysis, etc. So nowadays molecular diagnostic we are using as an effectively here to get very useful for as any conventional methods also like as any to do as in a serum, urine and different blood tests. Okay, the early detection is not possible. So when you are doing this one, early detection is not possible. Let's the recombinant DNA technology when you are using or DNA technology we are using here the polymerase chain reaction PCR and ELISA, that is enzyme linked immunosurveillance assay. So when we are using as any in these are some of the techniques that serve as any purpose of early diagnosis. So these are completely early to get you as any diagnosis, early to give as a result also. The presence of the pathogen like as bacteria, viruses, etc. So these pathogens is normally they're suspected only when they have pathogen has any the produced disease symptoms. Mostly they are complete there we are seeing that when the symptoms is present then only they are identify this a bacteria, this a virus. By this time the concentration of the pathogens is already very high in the body. However, very low concentration the bacteria or viruses at the time when the symptoms of the disease are not yet visible can be there detected by as an amplification of their DNA nucleic acid by PCR. So if in case body temperature is high, there is there showing as any they detect as any disease. If in case the body temperature is low, that time what happened? It is not detected as in the total diseases. So that time we are using an amplification the nucleic acid by using by PCR. So now can you explain how the PCR can be detected as very low amount of DNA. So when we are using as an PCR, see the PCR is now the routinely used as an detect as a HIV, the suspensed AIDS patients. So nowadays mostly we are using as an to early detection of the disease. The first one is what AIDS patients. It is being used as a detect as a maturation. The sorry, the mutation, the detect the mutation in genes the suspected cancer patients too. So AIDS patients, so as well as in their cancer patients, how do they are easily to get they are suspected. There is a powerful technique to identify the many other genetic disorders. So the PCR to get easily to get detect as in the many diseases. Okay, now that is next a single stranded DNA or RNA the targeted with as a radioactive molecule probe that is allow into as in a hybrid. It is complementary DNA to clone cells followed by as an detection using as an autoradiography. So when they are radioactive molecules probe allowed as a hybrid in a complementary clones. It is they get using by detection of using by what in a radioautography only radioautography. These clones having the mutated genes will be there hence not appear in the pathogenic film. So when the detected genes not appear as in a their photographic film because why here the probe will not have any complementary it has an mutated gene. So does not they are both are complement. So that purpose that genes are not detecting. Next, so that purpose which one they are choosing here the ELISA is based on the principle the antigen antibodies interaction. The infection by as in a pathogen can be detected as in a presence of the antigens, their proteins and glycoproteins, etc. So when you are using as an ELISA, it is to get easy to detection of as any the diseases or pathogens either in proteins as a glycoproteins, etc. Now by the detecting the antibiotic synthesized the agonist by the pathogen. So it is to get easily to get antibiotics produced against to as in a pathogen. Okay, now this is in completely molecular diagnostics. Transgenic animals. Animals that have had their DNA manipulated to process and express an extra gene known as transgenic animals. Means here any DNA to get manipulated and introduced into as any one animal and to get changed as the total modification, the total process we see in that process of transgenic animals. See the transgenic rats, rabbits, pigs, sheep, cow and fish have been produced all through over 95% of all existing transgenic animals or what? Mice. So these are completely examples actually. This best the existing transgenic animal is what? That is in mice. Why are these animals being produced? Because here they are completely the number of clinical trials did in these animals. How can be their benefits from such as any modifications? Let's try to explain some in a common reasons. So why mostly they get using? So they have in some complete reasons as there. Let's see one by one we discuss. First one is normal physiology and development. Normal physiology and development. See, the transgenic animals can be the specifically designed to allow the study of how to the genes are regulated and how they are affected on normal functions of the body and its development. So these are completely we can easily to get the study normal functions and as well as any what the characters they're easily to get identified. Let's say give an example. Study of complex factors involved in growth such as an insulin like growth factor. So for example in the insulin growth factor, how do they growing or they're completely in a organism body, you can easily to get study by introducing the genes from an order species that alter the formation of these factors, the studying the biological effects that result the information obtained about the in a biological role of in a factor in the body. So here first to get give as an introduced into as an gene into as an a body and what type of their factors will be the going on that clearly we discuss we are study in that first one is what that is in a normal physiology and development. Okay, this is the first one. Next, study of disease. The next one is study of disease. See, study of disease, the many transgenic animals are they are designed to the increase our understanding how to the genes contribute to the development of disease. So how to the genes that they contributed means how to they developed the enter the diseases. We can easily to get first study about the genes, how do they get controlled, how do they are increasing the diseases. First we complete the study and these are then specially made up of serve as any models for as a human disease that as an investigation the new treatment for the disease made the possibility. So how do that completely the disease will be the controlled. So what type of the treatment they require, the total we studied here only. Today, the transgenic models exist for as a many human diseases such as in a cancer, cystic fibrosis, and remediate the arthritis and their completely Alzheimer's. These are the complete the different diseases. The human disease also nowadays we are easily to get find that with the help of in this their transgenic genes. Okay, next third one biological products. Third one is what? Biological products. The medicine required to treat certain human diseases can contain the biological products, but such products are often the expensive make to make. So some biological products, for example, when he says the cancer, the medicine is present, that is so expensive. Like this kind of the total the expensive products also they are present. Next, the transgenic animals that produce useful biological products can be there created by as an introduction of the in a portion of the DNA which codes for as in a particular product such as any human protein that is alpha antitrypsin. It used treat as any that emphysema. So mostly that they mostly when you observe as in some any genes to get introduced into as any genes introduced into as any human or any organism, then what happened get increase as any that what immunity power or antibiotic resistant in your body. For example, like they given here particular product such as in the given here the human product that is in alpha 1 antitrypsin. It is used treat for as an emphysema. See, similarly, the attempts can be using made up of in a treatment of in a their phenylketonuria that is PKU, phenylketonuria. It is a cystic fibrosis. So this is a this also in some genes they are made up of to get treat and to control other diseases. In 1997, the first transgenic cow Rosie, the first transgenic cow is what? That is a Rosie. The produced human protein enriched milk that is 2,4 grams per liter. So Rosie cow to get to produce as an enriched milk. The milk contained as in a human the alpha lactalbumin, and it was nutritionally the more balanced product for as in human babies in a natural cow milk. So if in this milk to mostly to get help as in a kids or small babies, because they having as enriched nutrients as present in that cow they are producing. Next, after vaccine safety. Next, we discuss what that is vaccine safety. The transgenic mice are the begin developed use for in the testing of safety of vaccine before they are used as in humans. So first, directly we are not take action take the vaccines to humans. The first they are going as in some clinical trials as well as in some checking is very important. The transgenic mice being they used as a test of the safety of polio vaccine. So polio vaccine directly is not introduced into as in humans. First they did as in some clinical trials to as in mice. It's successful and found that in a the reliable to could be the replaced to use as in first monkeys and test the safety completely the batches of any vaccine. So after the mice, they get introduced into as in a completely monkeys, then only they get as a good result. Okay, next chemical safety testing. Next one is what? Chemical safety and testing. It is known as a toxicity safety testing. The produce is in a same that as a user testing toxicity of the drugs. So the drug capacity, drug that is toxicity or not, that entire thing first there easily their chemical safety is very important for that you as any organism. The transgenic animals are made that carry the genes which make more the sensitive toxic substance is non-transgenic animals. So if in case that is harmful, they get complete they are inhibited. It is prohibited to as in the disease, their medicine. If in case that is active, they're working. That is they complete their implement to as in any organism. They are in either mostly exposed to the toxic substance and the effects to studies. The toxicity the testing such as in animals to allow obtain to as a result in a less time. So when you're doing like this total chemical testing, they get give as a result within a short duration period. Okay, this is the incompletely in transgenic animals. Okay, remaining to discuss in next class. Thank you for watching. Please share, like, and subscribe. Thank you.